This video presentation will summarize my synthetic biology project, which is the mathematical modeling and simulating of the bacterial chemotaxis signaling pathway. Chemotaxis is a term used to describe the process by which bacteria facilitates movement towards attractants and away from repellents. In general, the process allows bacteria the ability to seek out favorable environments and avoid unfavorable ones. In many species of bacteria, including E. coli, transmembrane proteins detect extracellular signals and relay the information to the flagellar motors which dictate cellular movement. We will discuss the specifics of this pathway a little later, but what's important to know now is that the signaling pathway determines the direction of flagellum rotation, allowing the cell to dynamically alter movement depending on the environment. When the flagellum rotates counterclockwise, the flagellum forms a bundle of these fibers that propel the cell forward in a run. When these fibers rotate in the opposite direction, so clockwise, the fibers randomly disperse and induce a tumble bias in the cell. The chemotaxis pathway plays an important role in the development of multicellular organisms, the immune response, and cancer metastasis. By creating a model of the chemotaxis signaling pathway in E. coli, it may be better understood how the cells respond to chemical cues present in their environment. Experimenting with the different rate constants will allow for an analysis of those rates that have the greatest impact on the rate at which phosphorylated key Y turns on in the system. E. coli could then be engineered to alter these critical reaction rates to create highly sensitive bacteria that may allow for prolonged life cycles. This could be critically important in industries that utilize bioreactors and batch cultures, for the increase in sensitivity could alleviate some of the limitations on nutrient circulation. The chemotaxis signaling pathway is mediated through a transmembrane methyl-accepting chemotaxis protein, MCP. When repellents are present in the cell's environment, they bind to the receptor, causing the dimerization of MCP. Immediately following dimerization, the receptor recruits a protein called key A, which acts as a sensor kinase. That is, the protein is capable of autophosphorylation. It should be noted that key A autophosphorylates at a steady rate in the absence of attractants and the presence of repellents. After key A becomes activated by phosphorylation of its histidine residue, the protein activates another signaling protein, key Y, by transferring the phosphoryl group to the aspartate residue of key Y. Key A may also transfer its phosphoryl group to key B, a signaling protein that helps to reset the receptor to a pre-stimulus state, allowing E. coli extreme sensitivity to the environment. The activated key Y protein activates a 3-protein complex by binding the complex. This protein complex acts as the cell's flagellar motor switch. When activated by bound key Y, the complex signals the basal body to switch the direction of flagellum rotation from the default of counterclockwise rotation to clockwise rotation. Again, as mentioned, the clockwise rotation causes the flagellum to fan out, halting the forward motion of the cell. Key Y may then spontaneously dephosphorylate, or may lose its phosphoryl group to a phosphatase called key Z. The activation of the chemotaxis pathway may also be represented using logic gates. The production of phosphorylated key Y is the driving force behind reversing the rotation of the flagellum. Reversed flagellum rotation will occur in the absence of nutrients or in the presence of toxins. In other words, the cell will introduce a tumble bias when neither nutrients or toxins are present, or when toxins are present. In this example, it will be assumed that toxin presence is dominant over nutrient presence. There are some assumptions being made in the modeling of this system. It will be assumed the cell is placed into an unfavorable environment at time 0, meaning that there will be no prior stimuli causing an effect on the phosphorylation of key Y. Additionally, it will also be assumed that reactions involving the YZ complex occur very rapidly, allowing them to be ignored. This will mean Z is assumed to dephosphorylate Y at a constant rate. The reactions shown here outline the activation of the signaling cascade. K1 denotes the rate at which key A is autophosphorylated, and K2 describes the rate at which the phosphoryl group is transferred from key A to key Y. Similarly, K4 describes the rate of phosphotransfer from key A to key B. As mentioned, key Y may spontaneously dephosphorylate, as described by the third reaction, and all phosphorylated proteins degrade at a constant rate, shown by the bottom 3 reactions. The final reaction to mention is the dephosphorylation of key Y by key Z. and all reverse reactions are shown. We may then aggregate the effect of the different reactions on the concentration of different proteins in the system by forming compositors. For this model, 3 compositors would be analyzed, for phosphorylated key A, key Y, and key B. Of course, special attention would be paid to the phosphorylated key Y, for this is the driving force behind reversing the direction of flagellar rotation. MATLAB was used to simulate the proposed chemotaxis model and analyze the effect that the rates of phosphotransfer, autophosphorylation, and dephosphorylation have on the amount of phosphorylated key Y in the system immediately following signal activation. To start, the model was used to identify which rate, key 1 or key 2, has the greatest impact on the rate at which phosphorylated Y turns on in the system. 4 different conditions would be simulated using the model to answer the question about the role K1 and K2 were playing in the timing of the system following activation. The natural system would be simulated first with the values summarized in the table previously shown. Next, doubled K1 and K2 values were tested in an attempt to analyze the impact of these 2 different rates. As a reminder, K1 is the rate of autophosphorylation of key A, and K2 is the rate of phosphotransfer from key A to key Y. The final condition would look at doubling both rates. The following images of the simulation show the rise in concentrations of phosphorylated key A, key Y, and key B after the system was introduced to a toxin or after entering a nutrient-barren environment. Time in seconds is shown on the x-axis, and concentration in micromolar is shown on the y-axis. Key Y is shown in blue, and key A and key B are orange and yellow respectively. Based on these results, the model has demonstrated that the rate of autophosphorylation of key A has a greater impact on the concentration of phosphorylated key Y in the system than the rate of phosphoryl group transfer from key A to key Y. That is, the doubled K1 value demonstrated a faster response of the system than was demonstrated when the value of K2 was doubled. Figure B shows concentrations of phosphorylated key Y reaching 157 micromolar after 0.5 seconds. This is following signal activation, compared to 120 micromolar in the system with naturally occurring rates and 126 micromolar in the case of a doubled rate of phosphotransfer from key A to key Y. Additionally, the model demonstrated that increasing the rate of autophosphorylation of key A would allow for an increase in the amount of phosphorylated key B in the system. As previously discussed, key B is a signaling protein that helps to reset the receptor to a pre-stimuli state. Therefore, a rise in the concentration of phosphorylated key B would also increase the cell's sensitivity to the environment by ensuring the receptor is constantly adapting to the surrounding environment. This suggests that modification to the rate at which key A becomes phosphorylated may be a potentially lucrative target to increasing sensitivity of the cell to its environment. In an attempt to identify other possible targets for modulating the sensitivity of the system, the model was used to analyze the impact of altering the rate at which signaling molecule Z dephosphorylates key Y. 2 different conditions were simulated and compared against the naturally occurring system. The first condition looked at a doubled rate value for K5, and the second condition modeled a tenfold increase in this same value. It had been hypothesized that the concentrations of phosphorylated key Y and key A would decrease as a result of increasing the rate of dephosphorylation of key Y. It was shown in the figure that the concentration of phosphorylated key Y would drop from 120 micromolar after 0.5 seconds in the naturally occurring system to just 13 micromolar in this same time frame for a system with 10 times greater rate of dephosphorylation. However, it was also demonstrated that dephosphorylation of key Y by signaling molecule Z creates a negative feedback loop within the system. Therefore, by increasing the rate of this dephosphorylation, it is demonstrated that the steady state of the system is achieved much quicker. Genetically modifying the cell to demonstrate faster rates of dephosphorylation by molecule Z would allow for a highly sensitive and stable system so long as the threshold of phosphorylated key Y is still met to produce the desired output. In summary, the model suggested that the rate of autophosphorylation of key A and the rate of dephosphorylation of key Y by Z greatly impacts the rate in which the concentration of phosphorylated key Y increases in the cell.